Hi, I'm Dr. David Dobson. Welcome to Conversations. Today, my guest is Dr. Henry Atemnkeng, who has recently completed his PhD in biotechnology and biological sciences at the University of Southampton. His research specializes in agro-neuropharmacology and nematology and crop protection. Today, we will discuss his PhD work. Henry, it's a great pleasure to have you here.
I'm happy to be here, David. Thank you for the invite.
Henry, for people who are not familiar with nematodes in the soil, can you tell us what they are, why they are a global problem, and why managing them is so critical for food security?
Yeah, I'm very happy to. Basically, nematodes, for those who do not know what they are basically vermiform-like organisms. So they are worms, they are roundworms that live largely in the soil, and they range from different species. So there's free-living nematodes. So free-living means they feed on bacteria and other substances. And there is also the parasitic nematodes, like the plant parasitic nematodes, which are a huge problem. They cause troubles for agriculture production, causing issues like yield losses, which is really a problem. And these losses have been valued to reach 150 billion plus dollars annually. So they are really a big problem that need addressing and control in agriculture. But like I said, not all nematodes are bad because there are also free living nematodes.
Mm,
Yeah.
thank you. Henry, you have navigated an academic journey through Cameroon, Belgium, and the UK. How has this international experience shaped your approach to solving a global problem like nematodes control?
Yeah. So yeah, the academic journey has actually been a journey. So starting off from Cameroon, where I did my undergrad, I did a first master's degree in crop protection, and that's basically where I got introduced to nematodes because before then I didn't know what nematodes were. Um, and so I got introduced to these pests, and I started to dig deeper to find out ways to control these parasites. And that took me to Belgium, where I did a second master's in agro and environmental nematology, where I got to really understand the pest. Um, like the biology, the physiology of the pests. And then I got even much more interested, and I decided to carry it on to the PhD level, where I did a PhD in agro-neuropharmacology. And so basically my journey has been kind of like very tailored to a niche habitat or to a niche area, which is these nematodes. And for the PhD, I decided to go even niche. So I looked at a target receptor in this pest and how we can um, like understand how this receptor works in the pest and see how we can
Mm,
find ways to modulate the receptor and eventually control the pest. And so this was possible for me to do this because, first you need to be able to have the resource and the funding to do this kind of
mm,
research.
mm,
This is not the kind of research that I could be able to do home because first, the funding is not the infrastructure, the equipment. So
mm,
moving abroad and being exposed to I mean, having the funds and also the infrastructure to do the research that I did was just very um, yeah, it really helped me to navigate this research path that I am on.
wonderful. Very exciting journey you experience. Just curious, do you have like a farming background? Because
Oh,
obviously this is agriculture area you are
yeah.
studying.
I come from a very agrarian society. Cameroon is largely, almost everybody. If you're not doing it for commercial purposes, you're doing it probably for sustenance to keep the family running. So I come
Mm,
from that kind of background. Um, but also my under like I didn't say, but my undergraduate was in agriculture. So I did
mm,
plant health management
mm.
as the my specialty, but it was under the broad faculty of agriculture. So I do have an agriculture background.
Wonderful. That explains your love for this subject and this area. Henry, most people think worms as a nuisance, but to you, this tiny transparent roundworm,
Yeah,
scientifically known as, I'm going to try to say this word, Canthrabitis elegans,
yeah.
I mean, is that is the gold standard for your PhD research, right?
Mhm.
Why is this macroscopic soil worm so important to the future of crop protection?
Yeah, very good question. So this free living nematode, which you try to call or mention its name, Seniorabditis elegans, they're free living, so they do not cause trouble. They're non-parasitic. They
Mm,
pose no threat whatsoever. Actually, they're good for soil health and soil biodiversity because they help in breaking down organic matter in the soil, releasing nutrients for crops. So they're the good guys.
mm,
Um, we use them in the lab as models, or like you said, gold standard, because they for a number of reasons. First, their tiny nature makes it very easy for us, so we do not need like huge space to be able to work with them. Secondly, they have very fast reproductive life cycles, so they go from eggs to adults in only 3 days. And as a scientist, you want something that you can actually you can do like several experiments with them without being stressed for time. Another factor is they are transparent, so you can actually see internal structures of this organism in real time, which makes it very, very pleasant for some of the experiments that we do with them. And also just because they have a very good model because they have been completely sequenced, so we know what the genome of this organism looks like. And it makes it easy for us to do some of the genetic manipulations that we do with them. So we can take out genes, we can introduce genes, and study how this gene function or what a gene does in the organism, and it can inform us on how we can better control the pest. So they're really a gold standard.
mm,
We use them as models to study even human diseases,
mm.
and this is possible because we are all animals and we share some genes in common. And so what scientists do is they can look for corresponding genes and do what we call knockout and knock-in and be able to study these genes better. So they, like you said, the gold standard.
Excellent. Uh, just curious, Henry, we are just few months away from spring, a time to work on our gardens.
Yes.
If we were looking at a handful of healthy soil from a garden, let's say
Yep.
here in Canada, how many of these little roundworms might be there?
I didn't say this in the beginning, but nematodes are abundant in numbers. So
Okay.
it is arguably, so one fact about nematodes is they are the most abundant animals on Earth. So for every human, there's a corresponding 60 billion nematodes.
Oh,
That's how plentiful they are. But you cannot really appreciate this number because first they are microscopic and they live in the soil, so you do not even know that they're there. But believe me, they are there. So if you get like
okay.
a handful of soil, you get nematodes in their thousands,
Okay.
and they are going to be of different species. So fungivores, bacteriivores, parasites, all the different species just mixed. But what you also appreciate is, let's say, that's for, let's say if you got that in your garden. If you go in a commercial field where there is more like commercial agriculture happening,
Hmm.
the tendencies for you to find plant parasitic nematodes higher in number is also increased because if the host is present, which the host is the crops that you're planting, then there is this pressure, or the environment is conducive for these parasites to multiply. And so the tendency for finding the plant parasitic nematodes in commercial fields is even higher. But yeah, if you got a handful of soil from your garden, rest assured you find different species in their thousands.
Wow, wonderful. Very good. Uh, Henry, in your research at University of Southampton, you identified EAT2 receptor, a
Mhm.
kill switch that stops the parasite from feeding but leaves other animals unharmed.
Yeah.
Can you take us back to that aha moment in the lab when you first saw that optimized screen actually work?
Yeah, absolutely. So H2, this receptor is we in literature we know that this receptor is very important for the animal feeding. And we
Mhm.
know this because we've been able to take this worm, we knock out this gene called H2, and we see that the worm can no longer feed properly. And so this is just but that is not a control strategy. So for a control strategy, we wanted to find out if we expose these worms to these drugs that we're trying to find. These drugs can actually stop the worms from feeding. And if the drug stops the worm from feeding, it means the drug is actually interacting or disrupting receptor function. So a very aha moment in my research was when I introduced these worms to two drugs. First is an agonist called nicotine, and an antagonist. So an agonist is a drug that activates the receptor, and an antagonist is a drug that disactivates or blocks a receptor. And so using nicotine, I was able to activate the receptor. And with nicotine, you can overactivate the receptor and it becomes a problem. If you activate the receptor, it's good. But if you overactivate it, it becomes a problem. And so with nicotine, I was able to overactivate the receptor and block feeding and stop the worm from feeding, which was an aha moment because I wanted to show that I can use a drug to stop the worm from feeding. And that was exciting. And so I thought, okay, if that's the case, using an agonist, what happens if I use an antagonist? So a blocker of the receptor. And so I used mecamylamine, which is an antagonist. And I also showed that I could also stop feeding. And that was just very exciting for me because I've been able to show that I can in vivo target this receptor and bring about this physiological change, which is disrupting feeding in the animal. So that was some good finding. And with that, I was able to optimize the platform and screen also now a library of drugs to find other modulators of the receptor.
Wonderful. Thank you for sharing your aha moment, Henry. Henry, your research focused on targets that are clade specific. For our audience, how do you find a drug target that is lethal to parasite but completely harmless to beneficial organism like honeybee or human?
Yeah, very good question. And this is the problem that we face in crop protection, especially when it comes to pesticide control, because most of the pesticides are taking off shelves because they are not very specific how they act. They're mostly broad spectrum. I do not really want to be using broad spectrum pesticides because we have bees, birds, us ourselves. We do not want to be causing damages to us. And so one of the things that is very key to finding target selectivity is first of all identifying your target. And this is one thing that it too brought to my research because it is a unique target. When I say unique target is first to be able to show that this target is found in just the pest, I did what we call a phylogenetic analysis. And so phylogenetic analysis is basically trying to find a gene in other species. And I was able to show that this gene is not found in other animal species. So not in humans, not in bees, not in birds. It's found in just nematodes, which strongly suggests that the target could be a specific target, meaning that if I find drugs that interact with just this receptor, then there's a high degree that is going to be specific for just the parasite and not other beneficial organisms. That's how you go around these things.
Wonderful. Henry, a key part of your findings involves inhibiting perennial pumping.
Pharyngeal pumping.
Yeah. Why is targeting the way a worm eats a more sustainable strategy than traditional methods that might affect the whole nervous system of different species?
Targeting the way the worm feeds that was more like even a secondary response. That was not even the primary response. But again, if you target the way a parasite feeds, then because a parasite is a living organism, and like all living organisms, they need to feed to be able to progress with their life cycle. And so if you target that behavior, then it's basically KO, because if the animal cannot feed, it cannot grow, and it cannot reproduce. And so that was why we decided to look at ways to disrupt the feeding behavior of the animal. And so pharyngeal pumping basically is how the animal feeds. But again, that was not the primary focus. The primary focus was actually EAT2. And because EAT2 is a receptor which is unique to just nematodes, then we also realized that EAT2 again, it has a double function because it is unique in nematodes and it plays a very important role for the animal. So these are two important points. And so if we disrupt it too, then we stop the worm from eating, which is pharyngeal pumping. So it was more like yeah, a double win for us.
So you have conducted a very important applied research project, and I know that you are passionate about moving research finding from the lab to the real world. What are the
Absolutely.
biggest hurdles in turning your discovery into a product that a farmer can actually put into the soil?
Um, Good question. So the thing with research is sometimes in the lab or yeah, when you carry out these experiments in the lab, uh, first, is it cost? Is it what is it cost effective?
Mm,
Because the farmer does not only want to know that it kills the pest. He wants to know at what cost is he killing the pest?
mm,
Is it sustainable? And unfortunately, most of the drugs that I work with in the lab are actually expensive. But
mm,
that should not make science or scientists not do research.
mm,
What we can learn from that is we can study the structures of of potential heat compounds that we get and find ways to modify them and make it cheaper for eventual maybe field application. And also, secondly, sometimes the way compounds behave in the lab is not necessarily the way it's going to transpire in the field, because in the field there are other factors that this drug will have to go on, like environmental factors, yeah, like irrigation, maybe dryness and all that, which is or could actually seriously impact the how these drugs react in the field. So um, to be able to get to that level where we know that okay, this drug we can effectively use it in the field. We also need to do field trials to show that they actually have potencies in the field, like we found in the lab.
mm,
Also, try to make the cost um, affordable for farmers, basically.
mm.
Yeah,
Look like you have some more work to do, I guess.
absolutely. It never ends. It never ends. That's basically research.
Yeah, now you have completed your PhD. What is the next big thing for this research? Where do you see the field of agro-neuropharmacology heading?
Um, well, the good thing is we've been able to exploit new targets, and every day we're trying to find targets that are very specific for the pests. Um, and so, but we also realize that traditional ways of trying to find compounds can be lengthy and also laborious. So it takes a lot of time and yeah and energy. And so in the future, or the way I see it going in the future, is we're also trying to find ways to introduce like artificial intelligence,
Mm,
just so we can be able to cut down the time of the research and find novel compounds that we can use for these purposes as quick as possible and as many as possible. So yes, incorporating artificial intelligence into this research and also investigating more targets that we flag them as potentially selective targets.
I see a busy day ahead for you, Henry.
It is
Very
definitely.
good. Uh, Henry, for students who are looking at your journey and
Yes.
want to get into biotechnology or agro-neuropharmacology,
Mhm.
what is the piece of advice you would give them?
Um, so first, I would like to ask, I'll need to know what their motivation is, like, especially working with nematodes, because like I said, it's a very niche field.
Mm,
And so you need to be passionate about wanting to really pursue a career down this path, because it could literally take you to a niche that is very it's not broad enough. And if that's not what you're looking for, then I'd suggest maybe you stick with let's say working with the free living nematode like C. elegans, because with that it opens a little bit more options for you. But if you're going down the path with, like, say, plant parasitic nematodes, it becomes too niche. But first, I'd like to say your motivation. You need to be motivated. because getting to where I've been able to reach has been through motivation and a lot of hard work. Yeah, basically that. Just stay motivated and work.
Very good advice. Thank you for sharing your advice. Henry, thank you so much for joining us and sharing your research. We wish you the best with your next chapter.
It was an absolute pleasure talking with you, David. Thank you for the invite.